Hello everyone. So now we are going to continue the second KIMS in the biotechnology principle and then process. So in this we are going to study about the restriction enzyme. So what is the function of a restriction enzyme? So how this function going to be occur in a DNA. So in the first KIMS we learned that a restriction enzyme can able to cut the DNA strand. So in this KIMS we are going to study about how this restriction enzyme going to cut the DNA in a particular site or in a which form they are going to form this DNA pieces going to be studied in this KIMS. So now we are going to move on to the prerequisites. So the first one in the prerequisite is a restriction enzyme. So the enzyme which is used to cut the DNA strand which is called as a restriction enzyme. So another name for a restriction enzyme is a molecular scissors. So in this we are going to study about why they are called as a restriction enzyme. So that we are going to study in this KIMS. The next one is a palindrome. What is that palindrome? A word, a particular word. When we read it from a left to right or a right to left, they are going to give a same meaning. Let us take this word Hannah, a name Hannah. So when we are going to read it from a left to right, they are going to give a Hannah. So when you are going to read it from a right to left also, they are going to give the word Hannah. So this is a palindromic words. The next one is an elution. What is an elution process? So the elution process is nothing but in that elution process we are going to segregate one component from another component. So in this diagram it is like a separation of a DNA strand. So in this if you are going to separate a DNA from this test tube solution means that going to be called as a elution process. So the segregation of a one component from another one which is called as an elution process. Coming to the objectives, so in this we are going to study about the tools of genetic engineering. What are the different types of tools which is involved in a genetic engineering or a recombinant DNA technology. So some of the tools of a recombinant DNA technology is restriction enzyme, cloning vector, competent host. So these are the three important tools which is involved in a recombinant DNA technology. So first we are going to study about the restriction enzyme. In 1963, the scientists found out that there is a two enzyme which is responsible for restricting the growth of a bacteriophage in a E. coli cell. So they found out that in the E. coli cell that bacteriophage cannot be able to grow inside the E. coli cell. So they try to isolate the two enzymes. So in that one enzyme which is going to add a methyl group to the DNA. Whereas the another enzyme which is going to cut the DNA. So due to these two reasons, this bacteriophage cannot be able to grow inside the E. coli cells. So that enzyme which is used to cut the DNA, which is called as a restriction endonuclease. So this restriction endonuclease, the first restriction endonuclease which was identified is HIN2. So HIN2 is a restriction enzyme. So this was the first one identified in the restriction enzyme identification. So their function is depend on a specific DNA nucleotide sequence. So which was identified only after 5 years later. So initially they isolated this into molecule. So their function, the main important function is they are going to cut a DNA strand. That function has been identified after 5 years only. This HIN2, it was found that HIN2 always cuts a DNA molecule, but they are going to cut the DNA at a particular point, not at all the site. So they are going to recognize a specific sequence of a 6 base pairs. So for that 6 base pair which is called as a recognition sequence. So for a HIN2 molecule which is going to be a 6 base pairs. So likewise each restriction enzyme going to recognize a particular sequence in the DNA. So nearly there are 900 restriction enzymes that have been isolated from over 230 strains of an bacteria. So in the 230 strains of an bacteria, they have isolated 900 restriction enzyme. So each restriction enzyme has each recognition sequence. So that is why they exhibit that restriction enzyme. So that the enzyme which is restricted to that particular sequence only. So that is why they named it as a restriction enzyme. So each of which going to recognize a different recognition sequence. Now we are going to move on to the naming of a restriction enzyme. So how to name the restriction enzymes, on what basis they are giving a names. So some of the restriction enzymes like EcoR1, HIN3, BAMH1, SAL1, BAL1, HA3, SMA1. So these are the some of the restriction enzymes. So in that, let us take an example of an EcoR1. So in that we are going to split the word. So in this first letter which is going to be represent a genus, then second which is going to be represent a species. So these two represents from which this enzyme going to be isolated, from which prokaryotic cell this is going to be isolated. So for this E and then CO which represents the Escherichia coli. So this E going to be stands for Escherichia and then CO going to be stands for a coli, and then next that R going to represent the name of the strain, so which is going to represent the name of a strain. So for this they are going to represent the RY, and then the next one is a 1 which is going to represent the order in which the enzyme were isolated from the strain of a bacteria, on which order this restriction enzyme going to be isolated. So here it is going to be a 13. So Escherichia coli RY13 bacteria. From this bacteria we are going to isolate the EcoR1 restriction enzyme. So now this restriction enzyme belongs to a larger class of an enzyme which is called as a nucleases. So each enzyme going to belongs to the each classes. So in this restriction enzyme going to belong to the class which is called as a nucleases. So they are going to divide it into two. One is the exonucleases, another one is the endonucleases. So these two enzymes they are going to have a restriction property, that is they are going to recognize a particular sequence. So in this restriction exonuclease, they are going to remove the nucleotides from the ends of a DNA, that is either in a 5' or a 3' end. So they are going to remove the nucleotides in the ends of a DNA. But whereas this restriction endonuclease, they are going to make a cut at a specific position within the DNA. So they are going to cut the DNA inside the DNA. So that is why we are using this restriction endonuclease for our genetic engineering process or a recombinant process. So they are going to inspect the length of a DNA sequence, and then once they are going to find a specific recognition sequence, they are going to bind to the DNA. So once they are going to bind, they are going to cut the each of a two strands. So they are going to cut the each strand, that is the two strands in the DNA. So they are going to cut at a specific points in their sugar phosphate backbone. So they are going to cut initially they are going to cut the sugar phosphate backbone. So the sequence which is get recognized by a restriction enzyme which is called as a palindromic nucleotide sequence. So this is a pictorial representation of a restriction enzyme function. So let us take this is a vector DNA, this is a foreign DNA. So by using a restriction enzyme EcoR1, we are going to cut the DNA. So once we are cut, they are going to become a two different pieces. So this piece is going to be joined with the help of an enzyme DNA ligase. So once it is going to be joined, they become a recombinant DNA, because we are going to join the two different DNAs in a single strand, in a single DNA strand. So that is why it is called as a recombinant DNA. So in this, now we are going to move on to the palindrome. So always the restriction enzyme going to recognize a palindrome sequence. So what is a palindrome sequence? A group of letters that forms the same word when read both forward and then backward. So when a particular word, when you are going to read it from both forward and then backward, they are going to give a same word. Let us take an example word Malayalam. So when we are going to read it both forward and then backward, they are going to give the same meaning Malayalam. So likewise, if it is going to be in a DNA sequence, the sequence is going to be given same meaning when we are going to read it from a right to left or a left to right. So can you see this is a GAATTC. When we are going to read it in opposite direction also, they are going to give the same meaning, same type of a sequence. So that is why this sequence has been identified by the restriction enzyme, and then they can able to cut in the palindromic sequence. So they are going to read on the two strands at the same time, then only they can able to cut the DNAs. So how they are going to read means they are going to read in a 5' to 3' direction and then 3' to 5' direction. So once they found the palindromic sequence, they are going to cut the DNA strand. So how the DNA palindromic sequence inside the DNA means here should be a one sequence and then here should be a another sequence. So if it is going to recognize in this way only, they are going to cut the DNA strand. But when the same type of a sequence present in the same strand means they are not going to cut the DNA strand. So that one sequence should be in a one strand, another sequence should be in another strand. So then only they can able to cut the DNA. So when it is going to be in the same strand means which is going to be called as a mirror repeat. So when it is going to be in a two different strand means they are only called as a palindromic sequence. So when they are going to recognize this palindromic sequence, then they are going to cut the DNA strand. So during this DNA strand cutting process, they are going to produce a two type of an end. One is called as a sticky end, another one is called as a blunt end. So when echo 1, they are going to recognize a palindromic sequence. So this is a one palindromic sequence. So they are going to cut the DNA strand. So during that process, they are going to become a two separate pieces. So this type of a pieces which is called as a sticky end because they are going to make a L shape of a two pieces. So they will become a sticky end. Why they are called as a sticky end means they are going to form a simple hydrogen bond with their complementary cut counterparts. So that hydrogen bond can be easily formed when this piece can be overlap on this piece. So that is why they are very much easy to stick easily. So that is why it is called as a sticky end. So another type of end is called as a blunt end. So this sticky end facilitates the action of an enzyme DNA ligase to be more easily. So some of the enzymes which can able to produce a type of an end is given below. So in this you can see there is a HIN3, they are going to produce a sticky end. So PST1, they are going to produce a sticky end. So when a EcoRV, they are going to produce a blunt end. So this is how the blunt end will be, like a two separate pieces. So but whereas in a sticky end, they are going to be in a L-shaped pieces of a DNA strand. So this is how the sticky end and then blunt end will be. Can you see? This is a sticky end. So they are going to recognize a palindromic sequence. Once they are going to found the palindromic sequence, they are going to produce a overhangs of a complementary strand. So this can be easily placed over here and they can be easily sealed. But when it is going to be a blunt end, this sequence is going to be cut and then they are going to become a two different type of a blunt end. So in that there are going to be a no overhangs and then the edges will be uniform. This cannot be sealed that much easily, but this can be sealed that much easily with the help of a DNA ligase. So now we need to use a same type of a restriction enzyme to cut the both vector and then plasmid DNA. So this is a one important concept of using the restriction enzyme. So we need to use a same type of a restriction enzyme to cut both the vector and then foreign DNA and then um, host cell also. So we need to use a same type of a restriction enzyme. Then only they can able to produce a same type of an end. So when we are using here a restriction enzyme EcoR1 means here also we need to use a EcoR1. Then only the two pieces can be easily sealed. If it is going to be a BAMH1 here, if it is going to be a ECOR1 means we cannot able to seal easily. So that is why we need to use a same type of a restriction enzyme for the both vector and then foreign DNA. So let us take a one example. So this is a human cell. So this is a bacterial cell. So now we are going to isolate a one gene. Let us take an example of a growth hormone. So we are going to separate a growth hormone gene and then we are going to cut with the ECOR1. Now we are going to take a plasmid from a bacterial chromosome, so bacterial cell, and then we are going to cut with the ECOR1. So then only they can able to produce a same type of an end. So we can able to seal this growth hormone and then they become a recombinant DNA. So this recombinant DNA can be inserted into a host cell. So this cell can able to replicate more in a number and then this hormone can be produced more in a number. So that is why we are using a same type of a restriction enzyme to seal the two different species. When it is going to be an easy one means we need to use a same type of a restriction enzyme. So this is a process. So the restriction enzyme, they are going to cut the DNA strand. So the fragments have a single stranded overhangs. So this is a overhangs. So this can be easily placed and then with the help of a DNA ligase, we can able to seal the gap. Then the next important phenomena after the DNA fragment means we are going to separate and then isolate the DNA fragments, because we need to find out which one is the desired gene and then how much length it would be. So for that we are using a gel electrophoresis. So in the gel electrophoresis we are using a gel to separate the DNA according to their size. Let us take this is a setup. So the first one will be a sample. So this is a blood sample or a cell sample. So in this we are going to isolate the DNA and then once the DNA has been isolated, we are going to cut with the restriction enzyme. So once it is going to be cut into different fragments, so we are going to load it into a electrophoresis setup. So in this electrophoresis setup, the fragments going to be separated. How means the electricity going to be passed on to this gel. So as electricity passes, this DNA fragments going to be moved. So during that movement, that fragments going to be separated. Then we are going to see under a UV radiator. So yeah, under the UV radiator only we can able to see the DNA fragments. So in that the DNA should be loaded in a negatively charged side only. Why we need to load the DNA in a negatively charged side means DNA is a negatively charged molecule. So that is why we are loading the DNA in a negatively charged wells. Well is nothing but the place where the DNA going to be loaded. So this is we are going to load a sample in the wells. So once the electricity passes, they are going to transfer from a one end to another end. So they are going to move from a one end to the another end. So once they were moved, they are going to be separated according to their size. So in that, the shorted fragments are going to be moved very fastly inside the gel, whereas this longest fragments going to be moved very little far only. They cannot able to go that much far as like a shortest fragments. So based upon this, we can able to separate the DNA strand according to their size. So in this the gel which is used is called as a agarose gel. So agarose gel is nothing but which is a natural polymer extracted from a seaweed. So from the seaweed we are going to extract an agarose gel and then this agarose gel is used to separate the DNA according to their size. Now, so this is how the image under seen under the UV transilluminator. So then only we can able to see the DNA fragments. So once we need to see the DNA fragments after the passage of electricity means we need to take the gel and then we need to see it under a UV transilluminator. So how can we able to see under the transilluminator means we are using a ethidium bromide stain. So stain is we are going to color the DNA. So then only we can able to see it under the transilluminator. So that is why they are giving like a pinkish orange color. So that is going to be happen because of the ethidium bromide dye. So with the help of this dye only we can able to see the DNA strand. So if it is going to be this region going to be a gene of interest, means we can able to cut this region and then we can able to separate the DNA in this gel. So the separated bands of a DNA are cut out from the agarose gel and then extracted from the gel piece. So we are going to cut the agarose gel and then we are going to extract the gel piece. So that process is called as a elution process. So we are going to remove one component from another component. So this once we going to separate the DNA fragments, means they are going to be our gene of interest. So that we can able to use for a construction of a recombinant DNA by joining with them cloning vectors. So we will be studying what is the cloning vectors. So till now we learned about that they are going to be joined with the vectors. So you just remember that it is going to be joined to the vectors. Then this is a real image of a gel electrophoresis. So this is a negative side, this is going to be a positive side. Let us take this is going to be negative and then this is going to be a positive. So we are going to load a sample, that is we are going to load a gel here. So once we are going to load the gel, so once we are going to load the gel with the DNAs, the electricity is going to be passed. So during that electricity passage, this negative side DNA is going to be move on to the positive side. So once they are going to be move at a particular time, we can able to stop this and then we can able to see it under a UV transilluminator. So then we can able to cut the gene of interest and then we can by the elution process and then we can able to join with the vectors. They become a recombinant DNA. So this is a one example, like let us we need to find a relationship between a baby guppies with the mother and then father. So we are going to find out the similarities of a gene with the mother and then father with the baby guppies. So what happens means we are going to isolate the genome. So we are going to isolate the genome and then we are going to load the gene or DNA in this well. So once we are going to be inserted, we are going to pass the electricity. So once the electricity passes, the DNA is going to be moved. So in the shorter fragments going to be move for a longer distance, whereas the longer fragments going to be in a nearby place. So with this we can able to find out the similarities of genes with the mother and then father with the baby guppy. Likewise we can able to do it for the human also. So we can able to find out the relationship or we can able to find the length of a DNA and then we can able to go for a sequence also. So when we are going to separate the DNA according to their size, means we always use a gel electrophoresis. So coming to the question part, so the linking of an antibiotic resistant gene with the plasmid vector become possible with the help of dash endonuclease, DNA ligase, exonuclease or a DNA polymerase. You just kindly pause for a minute and then you can think and then you can resume a video. So the answer will be B, DNA ligase. The next one will be a restriction endonucleases dash, which is going to restrict the synthesis of a DNA inside the nucleus, or which is going to cut the DNA molecule at a specific site, or they are going to cut the DNA molecule randomly, or they are going to synthesize a DNA. So kindly pause for a minute and then you just think and then you can resume the video and then you can check for an answer. So the answer will be B. So restriction endonucleases, they are going to cut the DNA molecule at a specific site. So the next one, restriction endonuclease is also known as molecular glue or a DNA ligase or DNA polymerase or molecular scissors. So this is going to be called as a molecular scissors. So coming to the summary part, so in this knowledge imparting session KIMS, we have learned about what's a restriction enzyme, function of a restriction enzyme, and then gel electrophoresis, and then what is the use of a gel electrophoresis. So in an upcoming KIMS, we are going to continue the remaining concept. Thank you.